Assalamualaikum and hello everyone. My name is NURFARHANA binti FAISAL and today I will explain about the usage of DNA library in our biodiversity. There are many applications of DNA library by using DNA barcoding in biodiversity, and reasons why it is important are because of scientific discovery for human health and for the health of our planet. The DNA barcoding is a method used to identify species. It works by analyzing a specific region of DNA. This region is called the DNA barcode. The sequence of DNA barcode is then compared to a reference library which contains information of many species linked to their barcodes. The DNA can be sequenced by sending the amplified DNA to a sequencing service or by using a portable DNA sequencing machine. Firstly, for species discovery, the International Barcode of Life (IBO) project seeks to make DNA barcoding globally accessible for the discovery and identification of all multicellular life on Earth. In wildlife protection, DNA barcoding used by collection, extraction, sequencing, and translation of a species' DNA into a digital barcode and is being used to combat poaching and illegal wildlife trade. Investigators compared wildlife barcodes of evidence at crime scenes or confiscated animal products to the unknown barcode sequences of the suspected species. The Barcode of Wildlife project fosters collaboration among partner countries to build a global reference library that connects the work of researchers across the globe on a single platform and allow for a species barcode to be a public resource for biodiversity conservation, invasive species monitoring, and wildlife crime mitigation. Second, for detection of invasive species, DNA sample from the environment, eDNA, can be barcoded to monitor the presence of invasive species of concern. In DNA barcoding, scientists sequence a designated part of an organism genome and produce a barcode from it for a systematic comparison with the sequenced DNA of other closely related species. DNA barcodes are being developed on a wide range of plants and animals as part of its global effort to catalog the diversity of life on Earth. As an example, at the Invasive Insect Biocontrol and Behavior Laboratory in Maryland, entomologist Matthew Greenstone is using DNA barcodes in an unconventional way to identify insect predators best equipped to control the Colorado potato beetle. The Colorado potato beetle is the single most damaging insect pest of potatoes in the eastern United States. It also damages tomatoes and peppers and is known for developing resistance to any pesticides used to control it. Thirdly, for ecological assessment for conservation, which is important to understanding biodiversity and the effect of anthropogenic changes to make prediction for species survival and the planet's future. Example from a marine biodiversity. More than 70% of our planet is covered by oceans that have higher biodiversity compared to terrestrial or freshwater ecosystem. One of the problems that persists in the marine and estuarine habitat is the linking of the larval stages with the adult forms. DNA barcoding can accurately link the larval stages of species in order to unravel the life cycle of different marine species, which is usually difficult and in some cases not possible using the morphological approach. The threat of invasive species to marine biodiversity can be globally assessed through DNA barcoding. Fourth, DNA barcoding telling apart cryptic species. These are species that appear identical but genetically distinct, which can have important implications for biodiversity, wildlife management, and disease control. These species are difficult or impossible to identify based on their morphological traits, and they can be distinguished only with the use of biochemical or molecular methods. DNA barcoding is a highly useful method for identifying taxonomically difficult species. In recent years, quite a lot of new biorophyte species have been discovered by DNA barcoding. We know that DNA barcodes can aid conservation and research by assisting field workers in identifying species, by helping taxonomists determine species group, needing more detailed analysis, and by facilitating the recognition of the appropriate units and scale for conservation planning in our biodiversity. Thank you.